So Orchard uses a hematopoietic stem cell gene therapy platform. That platform is used across our investigational pipeline as well as the approved pipeline that includes Libeldy in Europe. The process is relatively straightforward. It's autologous. It means it starts with the patient's own hematopoietic stem and progenitor cells. Those cells are sent off to a manufacturing facility. And at that stage, then we insert a functional copy of the gene into those cells, and those cells are cryopreserved. They can be shipped anywhere at that point, and they're shipped back to the hospital where the cells are collected from typically. And then those cells are given back to the patient intravenously after that patient has undergone a conditioning regimen. So in that way, we're able to take cells that have a genetic deficiency in some enzyme or protein, correct that with a functional copy of the gene, and give those cells back to the patient. OTL-200 is also known as Libeldy, or it's also known as ARSA cell, it's shorthanded. The ARSA comes from Arsagene. That's the gene that is deficient in patients with metachromatic leukodystrophy. That's also the gene that's inserted into the construct that's used in that patient population. So in this case, we use a lentiviral vector, and in that we insert the copy of the cDNA, the ARSA gene, into this lentiviral vector, and then that vector is used in the cell. So what's important about neurodegenerative disorders is that when the cells, which are hematopoietic stem and progenitor cells, are given back to the patient, these stem and progenitor cells have the ability to traffic into the central nervous system, including the brain. In addition, those cells, essentially, because it's a lentiviral vector-based system, those cells have permanently integrated into them the functional copy of the gene. And so those cells can also differentiate into microglia. But importantly, the mechanism by which this particular gene works in this particular platform is to express that protein. That protein is expressed by those corrected cells. That expressed protein is picked up in the tissue and actually can then be picked up by neighboring cells that might not be corrected, like other neurons, for example. And so in this case, the important part of the mechanism of action is that those cells can traffic into the central nervous system. They produce the protein that's the deficient protein, and then that corrects the phenotype in this patient population, which would include a neurodegenerative phenotype.